So so it's definitely going to be a bit of a change of gear. So because probably many of you will not know me because I'm not doing anything related to synapses or anything like that. I'm a cognitive neuroscientist, but so at the risk of switching you all off now, I mostly look at human visual perception. But there was one project that we looked at that actually looks at sort of like short-term plasticity that I think could be interesting for you. And if not, it's just 2 minutes, right? So I'm going to be quick. So the thing that we were interested in is the fact that perception, or the inspiration for the project that I want to sort of quickly highlight, is the fact that perception is biased by a surprisingly long past. So when you present a stimulus and you ask people to report what they saw, then of course they can give a report. And then if you have basically sort of a uniform statistic, so there's no regularities in it, you will still see that your perception of the present is very much biased by what you saw before. Now, there's several components. There's like an attractive and a repulsive component. We think the repulsive component is particularly tied to sensation, so there's the sensory encoding of the stimulus. And that has sort of like it's spatially specific, so it's like retinotopically specific, and it has a very long tail, as you can see here. So you can see that the perception of a stimulus is sort of repelled by something that was shown like up to 8 stimuli ago. We were very surprised by that, because like independent contributions of 8 stimuli into the past, which at the time we didn't think really made sense. But there was like computational models that can sort of give an idea for like why that might be happening, some form of like efficient sensory coding. But the question that was inspired by this kind of behavioral data in the computational model, and so that's really the meat of my 2-minute presentation, is the question of like, does the sensory cortex indeed adjust to such long time scales? Could we sort of see sort of a correlate of sort of like a synaptic trace or something in sensory cortex that makes it kind of change its behavior to a sensory stimulus as a function of something that happened quite a long time ago? Now, I don't do mice, but luckily even cognitive neuroscientists nowadays can look at mice recordings by virtue of this beautiful Allen Institute data that was already mentioned in the previous talk. So what we did is we downloaded one of the NeuroPixels data sets from the Allen Observatory, where mice were basically doing something that was exactly what we needed, because they were shown these kind of gradings for 2 seconds with 1 second blanks. And then there were again sort of like uniform stimulus statistics. So the grading could be preceded by any other type of grading, or like 8 different orientations, and there was no structure to it. And that allowed us to look at like adaptation to sort of like when the stimulus was the same, for example, as the previous stimulus, or 2 stimuli ago, 3 stimuli ago, etc. And that's what we did in both thalamus and visually responsive regions. So here you see a description of all the regions that we looked at. And then there's a number of neurons we looked at this. So these are NeuroPixels data. So you can, obviously there is adaptation. So there's adaptation in the thalamus, there's adaptation in the cortex. This is stimulus-specific adaptation. So we really look here at the firing rate when the previous stimulus was the same versus the orthogonal stimulus. But now the interesting thing was that we saw this kind of stimulus-specific adaptation had a very, in the mouse at the encoding stage, so at the sort of like first 100 millisecond. It looked very much like the behavioral profile of adaptation that we saw in humans. So also in the mice, there's like up to, in this case, it's like about 30 seconds ago, a stimulus that was fairly briefly presented still had an effect, sort of an adaptation effect on the current stimulus. And that was really a cortical business. So we saw this long-term stimulus-specific adaptation in pretty much all the cortical areas. So here we're lumping over the cortex. Each dot here is a mouse. And you can see sort of long-term adaptation in the cortex, but not in the thalamus. There we only saw this kind of really short-lived adaptation. And we sort of speculate that, or well, what we think it means, but I really would love to hear also your thoughts on what actually is underlying this. But we thought that it means that these visual stimuli, even when you briefly present a visual stimulus, it leaves this kind of slowly decaying synaptic trace, maybe an eligibility trace, which has kind of consequences for how you encode stimuli, and which may be sort of like an automatic or sort of a moving history of the past that allows you sort of like the stimulus statistics of like the past minute, which may sort of help you encode the present more efficiently. That's all I had. it's probably more than a number of stimuli. So I didn't have time to go with that, but in the Allen Institute data set, they also have a different experiment where they have like 250 millisecond stimuli and no interval in between. So much more rapid stimulus protocol. And there we find similar behavior, particularly when we look at when we condition on previous stimuli. So I think it's really like indeed like a trace of like the past stimulation rather than time. Yeah. Well, it's more dramatic than that even. So here you have like a stimulus every 3 seconds. And there you have a stimulus every 250 milliseconds, like more than 10 times, about 12 times faster. So in that sense, I think, yeah, that those two data sets combined, we find pretty much the same effects in that other data set where we have much more rapid stimulation. So that's why we think it's stimuli rather than time.
I'm sorry, I just how do you think a synapse could remember? Like, what's the mechanism? Do you think there's some reverberatory activity, or do you think that there's something in the state of the synapse that has changed? And do you think it's local, or do you think it's happening in some distant location?
Right, well, that's exactly why I presented, because I have no idea about any of that. So I'd really love to hear your thoughts about it. So I'm not going to ridicule myself by trying to formulate an answer there. Yeah. But if you have any thoughts, I'd love to hear them. Yeah. Can be offline as well.
Right, maybe we can move to the second contribution, which is from Ben Wang, from my group.
Oh, yeah, hi, everyone. Yeah, I'm a PhD student from Jonathan's group, and today, yeah, I hope to share with you some recent modeling work we've been doing to connect a plasticity mechanism measured at single synapse level to a network. And the plasticity mechanism we were focusing on here is a calcium-based spike timing-dependent plasticity. And this plasticity is based on recent experimental observations that under physiological calcium level, the simple spike pairs will induce very little plasticity effect. And the main plasticity effects are induced by spike bursts or high pairing frequencies. And also it turns out these experimental observations can be captured in a calcium-based plasticity model. And this model has two types of calcium transient. And the fast calcium transient is triggered by individual synaptic spikes here. And the slow calcium transient is triggered by the co-activation of the pre and post synaptic neurons. And maybe more precisely, we actually have the equations for this different calcium transient. And as you can see, this slow calcium transient is triggered by some product term, which represent the co-activations. And when fitting to the experimental data, it turns out the decay time scale of the fast calcium transient is around 20 milliseconds, and the decay time scale for the slow calcium transient is around 100 milliseconds. But all of these time scales shown here are for a single isolated synapse. So a natural question we want to understand is what's the effect of this plasticity rule on a network level? Namely, can it create longer memory time on a network when taking into account the interactions between neurons and synapses? So we investigated this question in this recent paper, which just been published last year. And in the paper, we first formulated an analytical approximation to calculate the depression and potentiation region for this calcium-based STDP rule. And also, we found that the threshold between the depression and potentiation is modulated by the spike timing correlations between the spike trains. And this is due to the nonlinear slow calcium transient we introduced in the model. And then we investigated the induced memory timescales of these plasticity mechanisms on network by combining some mean field approximation and spiking network simulations. And in a network, each synapse, the strength of each synapse will undergo some fluctuations. And we define its the memory time of individual synapse as its first time to hit 0 weight. And moreover, so this is the memory time we are looking at on network level. And moreover, we also want to look at the memory time of the global network structures. But just for the memory time of these individual synapses in a network, we found that under the calcium-based STDP rule, the memory time of a synapse in a network depends strongly on its weight. And it turns out strong synapses tend to have long memory time and are protected, and weak synapses are forgotten quickly. And moreover, if the network initially has some cluster structure, then such calcium-based plasticity rule can maintain this cluster structure over hours, despite that the calcium transients on individual synapses has a decay time scale around hundreds of milliseconds. Also, we further shown in the paper that the nonlinear slow calcium transient in the model is essential to generate the stability of strong synapses and the stability of the network structure shown here. So in summary, I just want to summarize a little bit. So here we developed a mathematical framework to connect the plasticity mechanism measured on cellular level to its effect on the memory timescales on the circuit level. And here we focused specifically on that calcium-based STDP rule. But in the future, it will be very interesting to see whether similar formalism can be used to understand other plasticity mechanisms measured on the cellular level. For example, the behavioral timescale synaptic plasticity discussed in the earlier presentations, and understand their effect on the circuit level. And hopefully, so this work will give us a better understanding about how the memory timescales on the behavioral level is generated and what's the mechanistic link between the cellular mechanism to the behavior.
I'm just curious whether your model can explain Torres's work from the previous talk.
Which talk specifically?
The one that we just had right before yours.
Oh, the SST? Oh, no. Yeah, so that, yeah, that will require some maybe discussion after the presentation.
The first time scale here is 100 milliseconds,
Yes.
which there's a strong requirement for coincident firing of painful spike within a very narrow time window. If that happens, then there's a strong time scale of about 100 milliseconds that was matched to some experiments. People talk here about much longer time scales of seconds, both in Floristock but also in Crispin. So our current model cannot explain these observations, but because the language of the requirement for synaptic plasticity in these experiments was elevated calcium through calcium plateau potentials, we think that's why Ben said that the same formalism might be extendable to these more new forms.
Also, in some sense, our model is just a model with multiple eligibility trees with different decay time scales. So in principle, we can just use a new decay time scale and plug that into our model and understand their effect. So that may explain this more slow synaptic plasticity with wider time windows.
So maybe we can open the floor for any questions to any of the speakers or general discussion.
know if this is working. Anyways, I wanted to just bring up for discussion something about Jen's talk, and this is a question to Jen and the audience. I mean, I really think it's absolutely foundational, this idea, and tell me if I'm summarizing okay, this learning of the learning rules that we have going on. And this is something I don't think we consider enough in the computational field. And what should we consider in the computational field in terms of how valuable that would be if our learning rules themselves would be plastic? And do you see this operating in the neocortex? I mean, I do wonder about the cerebellum being a bit more specialized. You know, the feedback times. Does that happen in the cortex? And this is just a general question for computational people in Jen.
Yeah, well, I can offer
I don't know if this is on.
maybe we could use this one up here. Is this working? Maybe we can use this and you can pass those. Yeah, so this is a question that I'm very interested in, and if anybody has thoughts of like, where could we go, which other synapses could we go looking for this kind of temporal metaplasticity, or how to find it, I would be very keen to know and collaborate and or just like cheer you on. But you know, I think it's a fundamental question. Is this a unique property of these synapses? They have this climbing fiber input. Does that make it somehow special and unique? And maybe the parallel fibers do sparse coding. That's one idea that's been out there, and that would help this work. Although I think you could also do it with a threshold on the parallel fiber. But I can imagine that only these synapses do this, and that might be the reason then you have a cerebellum, is to do this kind of temporal metaplasticity, to learn about the timing statistics between events in the world, and maybe then the cerebellum helps other parts of the brain learn because of that. But I doubt that. I would have to bet other synapses can do it, because if you are doing any kind of association, which sound goes with which visual input coming into this particular place at the brain, those auditory and visual stimuli are going to tend to arrive at a given location in the brain at a particular interval. At one site, it might be a 30 millisecond delay for the visual input to get there, and somewhere else the visual delay might be longer because you're further along in the processing. And knowing when two things that go together arrive at a given site in the brain will help you to learn which things to associate, right? Because you've got two streams coming in, and learning the statistics of when they tend to be correlated could help you learn specific pairings faster. So in the break, Manish was saying, well, this temporal credit assignment problem, aren't you just putting it off? Yes, but maybe the metaplasticity learns that temporal credit assignment over longer statistical sampling, so that you can learn rapidly with a few samples which of two inputs coming to a site should be associated. So I have to bet that other synapses can do this, and I hope we can find it.
Just given the challenge of the model. So in fact, there's a body of work on what's called method gradient reinforcement learning, whereby they have black box techniques and white box techniques. The black box techniques just say, let's have a system which actually learns a whole collection of any sort of plasticity for itself, and can generate new targets, can generate all sorts of things in the service of doing reinforcement learning. And then white box techniques take existing learning rules and then manipulate the parameters of those learning rules, just like Jen is showing, in order to improve learning. So there's actually quite a lot of there's some interesting work along those lines.
So in that case, you're improving learning.
Can you use that? Is this what it's recording?
Sure. In that case, correct me if I'm wrong, Peter, they're selecting or developing learning rules that are used to train networks across different networks. So it's not really online, like is it? Like Jennifer's is saying?
So the issue of what you call a single task or multiple tasks becomes a bit complicated in that case, right? So if you say I just have one long task, just like I think it would be in Jen's case, where you just you're experiencing dock rearing, and that's just one sort of task. That's sort of setting some aspects of the learning rule. Or you have a set of environments where you keep on one environment after the next, and then you're doing this meta learning. So in fact, people have done both. And so I think it is aspects of that. But of course, this is in a very, they're not trying to look at individual synapses in quite the way that this beautiful detail that we see.
And what's the result of those studies? So do they find similar rules with different time constants? So Jennifer has this beautiful case where it shifts. It's primarily a shift in the valley, and she's calling it the temporal metaplasticity. But you could imagine very different forms of learning to learn in which the direction of the synaptic plasticity changes, or instead of having a symmetric STDP curve, you now have an asymmetric STDP. So what's the result of these?
So as I said, people have looked at them in much more abstract domains where, the question is whether you can, warm up Iceland to get an Atari game to learn a bit faster. And so that's essentially the context in which they've done it. And so then they haven't looked so much, it's very hard then to look at the individual details. But I know that, for instance, people like, there's work from Maria Eckstein where she's doing descriptively and say, let's look at behavioral learning in humans, for instance, and then instead of taking a regular reinforcement learning rule and saying just fit a few parameters of it, treat that as being something which is parameterized as a neural network, and then learn the best ways of that neural network. So in some sense, that would then be the optimized, that would be capturing what sort of plasticity people would have the access to. So that's another way of getting at the same sort of question from a descriptive rather than mechanistic point of view.
I had a follow-up question to Jen related to this discussion. It seems like your data suggests that the entity responsible for setting the plasticity rule is the neuron, because the time scale you suggested it's the same for all the synapses of the same Purkinje cell. So can we somehow use that as a constraint on the mechanism that implements this metaplasticity if we know that the entity that's in charge is the neuron and not like the synapse or the dendrite?
Yeah, so we don't know whether the rule is tuned like for all of the parallel fiber synapses onto a given postsynaptic neuron. I was saying in the question period, I hypothesize that's the case because the delay that matters is parallel fiber made the Purkinje cell fire when it shouldn't. That caused a behavioral output that was bad, and there was an error signal that came back. And so that loop should be consistent for, my first order hypothesis is that loop should be pretty consistent for all the parallel fibers. It's not necessarily the case though, as I've thought about it more. So maybe in a certain context, say low contrast, the visual input comes back later. And so if some parallel fibers are active during low contrast and some are active during high contrast, you can imagine two sets of inputs that get feedback at different delays. So maybe you do want to do it in a synapse specific way. So we could try to figure out how to test for synapse specificity, especially if we could induce the metaplasticity in the slice, then we could do that. In vivo, it takes a long time, but that long time of a few weeks to do the metaplasticity may be because it's sampling and trying to pick a small peak in the distribution out of many irrelevant parallel fiber climbing fiber intervals. So that's another question. With concentrated input at a certain interval, could we tune the plasticity mechanism faster?
I have a question for Christine. I'm sure it's just me not understanding, but at the start you contrasted Hebbian plasticity with behavioral timescale plasticity. But I wasn't clear how the behavioral timescale plasticity you were presenting couldn't be explained by Hebbian plasticity. I mean, you're getting depolarization, there must be interacting with some activity. We've got longer time frames over which this may be happening. So I wasn't clear why you were making that distinction particularly.
I mean, okay, so to me, Hebbian plasticity means basically those synapses that drive the action potential will only those synapses will undergo potentiation because, what wires together fires together. So in the BTSP, it's not like that. Yes, there is also association between some things, but it's not association between input and output. It's association between a dendritic plateau potential that is driven by one type of input and another input, right? So in that sense, the concept is a little bit different than in Hebbian plasticity. Does this make sense? Or it's not a pairing between input and output. It's between two different types of signals in the cell that drives the plasticity. So in that sense, it's to me conceptually something different.
Yeah, I guess in the great scheme of Hebbian plasticity, in my mind, it's very similar. The postsynaptic cell has to fire for a reason, and in the Hebbian postulate, cells are working together to repeatedly fire the postsynaptic cell, and doesn't say where those inputs are that are doing this. So yeah, you have SLM activity, you have stratum radiatum activity, let's say, and they're generating postsynaptic activity. So I mean, to me, it's still quite Hebbian in nature, and just the way I would look at it, I guess. Yeah.
Sure.
Not that it really matters, I suppose.
To me, it's something very different. But,
Can I add to that, Christine? Yeah.
Sure.
I mean, I think by Hebb's own definition, when neuron A takes part in firing neuron B, which is not the case in Christine's case, right?
No.
So neuron A is not what's causing the firing of neuron B because it's the plateau potential from the EC3. So by that definition, I would say that they are fundamental. The causality is not there. But I understand there's still a correlation. But I think the causality is what's missing.
I mean, I think if you know.
So I'll add another view to that same topic, and that is, could it be that the setting of an eligibility trace is Hebbian in nature, where you've got pre and postsynaptic activity, and that meets the requirement for turning on and generating an eligibility trace, and then the dendritic plateau potential is sort of gating mechanism, it's an enabling mechanism.
I mean, I think
we're talking about three-factor.
Three-factor, you're describing three-factor rule, but I will say that the eligibility trace is in synapses that do not cause any action potential firing, right? Because in this lab that, that presumably the plateau happens and plasticity happens, none of these synapses, especially the ones that are active seconds before, causes any action potentials, nor in the labs before. So I think, yes, if you interpret HAB as just postsynaptic depolarization in a synapse, yes, sure, then we could, but it's certainly not causing action potentials, right? So in that sense, this credit assignment that HAB postulates is not the case in BTSP, I think. We don't know also, I mean, maybe kind of expanding a little bit on that, we don't know exactly what pattern causes an eligibility trace to form, right? Does it need to be, is it really just input? Does it need clustered input? Does it need a small dendritic spike, right? All these things we don't know. I mean, BTS, you mean like a change in the release probabilities, something like that? So, I mean, I think the only thing that we've done to test that is that we looked at pulse facilitation before and after. It didn't change. This is a very coarse measure, of course. So these are certainly experiments, I think, that need to be done to better understand what's going on.
I have a question for Allison and also the whole group that continues a discussion we were having at lunch. So when you finish your decades-long effort and you have this description of like changes kind of flowing through the circuit, then what? Like, how do we get from there to a concept? Is there a concept? And this is a reaction to your work and some of the things we heard this morning, and just, how do we deal with the complexity of all of the different mechanisms that are there in the circuit, even for very simple kinds of learning that are very close to the input layer, and then to get from there to more complex, But even for the simple forms of learning, we have this
daunting complexity.
Well, the first thing I would say is that when you make an association, right, just changing, you could stimulate the whisker and drive a behavior. You could stimulate the trigeminal and drive the behavior. You could stimulate the thalamus and drive the behavior. You could stimulate sensory cortex and drive the behavior. Doesn't mean that's where the memory lies, right? So it's the memory is there's an association between the stimulus and then some behavioral response or then some expected outcome. So it seems like it is quite reasonably not in sensory cortex. So there are sort of two parts of this. You know, one is maybe, and I'm paraphrasing a little bit, like why study sensory cortex at all? Part of the reason is that we know something about the circuitry that's involved, and we can then start to make some inferences about what might be important. For example, you know, we were looking at the apical dendrite. We were looking at synapses onto, so these thalamocortical synapses are on the, you know, apical tufts in layer 1, and, you know, we know these synapses are getting stronger. Are those the ones that are getting stronger? And for those inputs onto layer 5 pyramidal cells, no, they're not. They're not. So, right, it helps us then, you know, begin to test some models that we have about how some of these lower level things might be implemented in a more complex circuit during an actual behavior. So no, I don't think the memory necessarily will lie in sensory cortex, but there's a lot of things that we can rule out or, you know, test because of the plasticity that we're seeing. And I guess the other thing I would say from a personal standpoint is, you know, as a scientist, your job is to kind of keep your eyes open and, you know, to try to make sense of the observations that you have. And this has just been a thrilling ride of, you know, things that we didn't expect to see. And then, you know, like the identification of a few variables that we can start to now manipulate in very precise ways and, you know, get and see that the system is detecting, noticing those things. And some of it is outside of any theory that we had, which, you know, that's why it's thrilling, right? Because, you know, we're wrong. So yeah, the end goal is, right, to be wrong. That's our job, is to be wrong, right? To put yourself to test something hard enough that you're not right at the end of it. That's how you move forward.
Alison, beautiful words. Actually, this was almost like closing words of a session, so I don't dare to add to that. But I think brilliant data you had been presenting, but I think also something that may help to dissociate different forms of learning is to consider the instructive nature versus the permissive nature of plasticity. So I think something. So I think in your experiments you're brilliant in teasing out class differences in the statistics, right? So, but you could imagine that there are also changes which are actually not changing the overall statistics by comparing two groups, so that patterns are changing, but comparing two groups you wouldn't be able to see it. So I think I agree it's a very daunting task, and maybe trying to get to the goal would be, for example, making this dissociation between permissive forms of plasticity and instructive forms of plasticity, which actually really store the information.
Do you mean permissive, like these things have to happen in order for the association to happen somewhere else?
Exactly.
Yeah. I would say it's likely that that's the case, although this is a very simple form of learning. And I have a great colleague at Carnegie Mellon, Kate Hong, who has, you know, her work's shown that if you get rid of sensory cortex, the animals can learn just fine. You know, she and I actually have a grant together. I don't think that there's a particular contradiction there. I think if you don't have it, you probably use something else. But if you do have it, then you can use it, and then perhaps it is permissive in some respect. So, yeah.